Hello, I'm Dr. Ellie Jameson, and I'm going to be talking to you today about DNA. So we're going to do the science session all about DNA. So what is DNA? Where is it? And why is it important? So DNA, the long name for DNA, those three letters, is deoxyribose nucleic acid. And this is the instruction manual of how to make living things. So it's inside every living cell and inside viruses too. And it's a really long, thin molecule, and it sort of looks a bit like a ladder that's been twisted round. So it's got two bits up the side and these rungs in the middle, and those rungs in the middle are the important bits which encode the whole of what's inside every cell. So for each of these rungs of the ladder, it can be one of 4 different bases. So these 4 different bases make up the DNA, and depending on what order they're in, they can tell it to make all the different parts of a cell, and whole animals and plants and all the organisms on Earth. So without DNA, we wouldn't be able to repair ourselves, make new cells, make babies, make new things. So DNA is really important to tell cells how to remake themselves and what they should look like. So in animals and plants and big organisms, usually what happens is that they combine, 2 adults will combine the DNA from their cells to make a new baby or a seedling or plants and animals. So they mix them together, so each new baby will be unique. And things like bacteria don't do that. They can make new ones of themselves by just copying all of the DNA that they've got inside their cells and then making a new cell from that. So that's splitting one cell into 2 new daughter cells. It may sound a bit boring for the bacteria that they don't have to combine their DNA with everything else, but with bacteria, they can do that at any point while they're alive. They can take new bits of DNA and put it into their DNA. So they can combine at different points, but not to make a new cell. So without DNA, they wouldn't be able to make these new cells either. So it's important to the bacteria to be able to make new cells. So inside our bodies, we've got lots of cells inside us, but also we've got lots of bacterial cells as well as our own human cells. So we've got actually 10 times as much DNA from all these bacterial cells as we have from our human cells. So that means more of our genetic code inside of human bodies is bacterial DNA rather than human DNA. And that's all those bacteria which help to break up your food and give you the nutrients you need from your food. So they're really important to us. And so even if they're not human cells, it's still part of us and still good. So we've got lots of DNA inside of us. And if you take the DNA out of one cell inside your body, you'd be able to stretch it out to about 2 meters long. And if you took the DNA out from all the different cells inside your whole body, it would stretch across a solar system twice, so 2 times across our solar system. So our solar system is just our sun and the planets that circulate around us. So we've got enough DNA inside one human, because it's very, very thin, but can be stretched very, very long to go twice across our solar system. So I'm interested and I work with viruses. So viruses are really just a piece of DNA or something else called RNA, which is a similar molecule, wrapped up inside protein. So this protein covering is called a capsid. So viruses are just really a piece of DNA wrapped up in proteins. So quite simple, which means they don't have the machinery to make more of themselves. They've got the genetic code in there, but they can't use it to make more of themselves. So viruses need a host. So this is a cell that they can go inside, they can put their DNA inside, and that cell will make more viruses for it. So that's why viruses can cause disease in us, because they can put their DNA inside our cells. They can use our cells to make more viruses instead of making more of our cells to repair ourselves. And that can be bad for us. But what I'm interested in is bacteriophages. So these are viruses which infect bacteria, so they're called phage. So these phage can kill bacteria which are bad for us, and we can use them to help us. So they use the bacterial cell to make more of themselves, and by doing that, they, instead of just making us sick like they would inside a human, they kill the bacteria, because bacteria is just a single cell. So it's bad for the bacteria, but if the bacteria is bad for us, then actually viruses can be our friends as well. So we can use these viruses for what we need. And I use these phages to try and kill harmful bacteria. But what I need to know before I can use these phages to kill harmful bacteria is there's nothing inside the phage itself which could harm us. So I need to check inside these phages' DNA and I need to sequence the DNA inside these phages to see if they have any toxins that they can make, or if they have anything in which can stop antibiotics from working. So this is called antibiotic resistance genes. So if they have any genes inside the viruses for toxins and antibiotic resistance, then I can't use these to kill off harmful bacteria. So what I need to do is, first of all, I need to get the DNA out from inside my phages, and then I need to send it off for someone to read it. So let's sequence it to see what this string of bases inside is telling it to make. So to show you how we do this extraction of the DNA, we're going to use kiwi fruits.